Well, thank you very much for having me here. Since I'm kind of a newcomer to this bioinformatics, I will take a few minutes to introduce my group to tell you where we come from. So I'm in statistics and EECS. My people come from actually both fields, statistics and EE, and Siqi, who's there, and Carl from my group. And this collaboration started about 4 years ago when Ben Brown, who was part of ENCODE, approached me, suggested collaboration with Irvin, who is a biologist at BL, and Peter Berkeley was the one who already made connection with Brown and Haiyan Huang too. So I'm kind of riding on their connection and working on this. And so my group really, for the last 15, 20 years, engaged in high-dimensional statistics, machine learning, did a lot of sparse modeling theory. And you can see that we're actually using that for this problem in big time. I have been also engaged in remote sensing and computational neuroscience. So we also borrow idea from there too. So when we started this, we actually had a lot of experience, but not so much in genomics. Although 15 years ago, so I did do a little bit physical mapping. That's why I heard of Mike's work. And then quickly I disappeared because it was so technology driven. And without good collaborators, I went to neuroscience. So now with the good collaboration, kind of coming back. So that's how my group works. And we have theory people, we have algorithm people, and also people like Siqi spend half of their time in the wet lab. So this is the team. It's one of the projects I would call, I think one of my favorite projects now is really data science. We have 3 teams of people. This is the IBR group co-lead Irvin, Siqi Wu, who is a graduate student, basically co-advised by Irvin, who's sitting in the back, who answer all the hard questions, and my team. And Sue Staniker's lab is doing NALCA experiments. And we have a team at Tsinghua University, computer scientists, try to help us scale up the computation based on Spark and Fiji. So it's really 3 teams working together. And some of you actually have probably know about this data model or have worked on that more than I have. Owen is here, who actually worked on this earlier than we did. So this is a public data set produced by Sue Staniker's group, the Drosophila group at the IBL. Instead of different conditions, experiment condition, different tissues, it's kind of different tissue here too. We have gene expression data in situ kind of embryo. This is Drosophila embryo, about called stage 4 or 6 or 2 or 3 hours. And there are other available data as well to give the spatial information. And also you can kind of think of this location. It's kind of like different conditions on RNA-seq. Because we also try some data, it seemed to make sense too. So primarily, we developed this platform, hoping to have other people also use it to support this like enriched spatial or gene expression data. And the way we work is that we usually take a problem. I mean, we're kind of statisticians, so we like general methods. But we really drill into a problem and work with biologists and validate it, and then we kind of come out as a general methodology for possible other problems. So we first finish the first phase, which is 4 years. We have first paper coming out, is to really validate what we do, feel way comfortable to publish. And this is known to everybody here, but I just want to point out, I don't have different experiments, conditions, but think of my pixel location as like different condition. You can think of it as like a different tissue too, because there are different organs there, but it's all in situ in the embryo. And this is a microscopic image, which is kind of you have a lot of processing, huge amount of image processing work done before we got on the problem. And I'm not going to talk about that because it's mainly built on other people's work. And Drosophila, for those of you who probably all heard of, Drosophila, the Drosophila people I work with tell me that share 75% of genes with humans. And yesterday I heard from Donna that actually the diabetes, people have been studying diabetes using Drosophila model. And I also got some paper from Sue last night. So hopefully it's a relatively inexpensive model to study a lot of basic mechanism, which hopefully will generalize across to higher beings. And the data we're looking at is 1 terabyte. They tell me they have most of the genes, 7,000. And we kind of set up, when we started the project, to see whether we can reproduce this fate map, which was obtained by laser ablation old technology, try to cut things out to see which part is which. This is different. This is kind of, the purple is like a neural region, and this is more like gut, foregut, things like that. So we say, can we use our data, modern data, high throughput, to figure out the different regions? Can we figure out these two genes interact or not? For me, interaction in this talk is about co-occurrence at a co-variant. It's not just co-occurrence, co-variant in a particular spatial location and region. And I can see from other talks that there can be different things. And the genes I'll be talking about today, about 700 of them, mostly transcription factors. Because for the early stage, based on biology, that most of the genes correspond to transcription factors, and there's a lot of cascading of reactions happening. They will work, as I said, this data set is existed and still being added on for at least 5-7 years. And the lab themselves publish papers on human annotated data, basically look at the morphology of the tissues, and then say which region is that. And a lot of work, including Irvin's work, use that to do supervised learning. Okay, so we will go back to supervised learning as a validation, but in the beginning we're more doing unsupervised learning in a meaningful way. So one thing I think the design principle for the lab was spatial information is rich and heterogeneous. Can we take advantage of that to figure out local gene and gene networks? One prominent example of spatial information useful for gene-gene interaction is the famous gap gene network. This German scientist got the Nobel Prize in the 90s for her work in the 70s. Most of the Drosophila genes were German because her lab discovered many of them. This means giant. So this is like a schematic embryo. The giant gene is here and also here. It says that giant means that if you knock out this gene, the embryo will become big or giant. And this is tailless. If you knock out this gene, the embryo becomes tailless. And these two means the same gene, and they play different roles at different locations. And in the anterior part, interact with croupo, and here interact with knee and tailless and hunchback. So this says that depends on where the gene is. I mean, I'm not really a biologist, so I try to explain to a statistician in a way that the genes are like workers. For the embryo development stage, the workers team up to build different houses. The different houses are different organs. So that's what they do. This is the fruit fly embryo. You can see the segmentation. The gap gene network is instrumental for building these vertical divisions. And this is the human embryo. You also have the segmentation. So that just gives you a sense that there's things to learn from to bootstrap up to humans. And we did a lot of processing to go to 405 superpixels so that we have a data matrix we can work with. And there's using the work from Eric Singh's group from CMU to put them on the same ellipsoid, so a lot of work was done. So that's the data we work with in the talk. And just to show you a hunchback and giant, we know from the gap gene network, they kind of repressive each other. Look at giant, look at hunchback. You probably can visually move this piece here, and they fit pretty well. So they are repressive of each other. This is to show you that our data does have information to recover the gap gene network, right? Because the spatial contingency. And this is another work to show that in the Drosophila literature, people are already thinking about segregation or segmentation for the embryo, because different organs will grow out of different parts of the body. And this is just another more recent work than the one I show you, fate map on the building block. So this is one slide of overview of the teamwork, which is 3 teams coming together. The first step we did is something we borrowed from computational neuroscience. Later we went to non-negative matrix factorization. And that is decomposition. And then we can build local gene networks. And then we do recommendation for the lab. We have some early result on knockout experiments I will share a little bit with you. And then we feedback loop and go back to do more analysis and use Spark and Fiji, both open source, to build the whole platform. So that is an iterative process of wildlife people. We're kind of in the middle, the statistician. Kind of computational, but need help from computer science to do the really scaling up. And if, in terms of methodology, the contribution of this work I'm talking about, it really came from the stability which I've been advocating in the statistics and machine learning literature, which I've seen in many talks, people already doing that. You perturb your data in a sensible way, and you want your conclusion to be stable. And it's very easy to do, and I think we can do at many levels. When I teach applied statistics PhD level course, I actually tell people for classroom, because it depends on which one you use, you end up with different rights. You should have conclusions robust to the different methods you choose. So, so kind of this paper is kind of overview paper tying together some works from my group and with my collaborators to pose that reproducibility, the first step, which is easy to do, is this sensible perturbation. Could be adding noise, could be adding synthetic data, could be just bootstrap subsampling, but it has to be context appropriate. And that's what we did here, is actually a robustness relative to random starting point for a non-convex optimization problem, and worked really well. So our methodology contribution, I said, we call STAT for stability, non-negative matrix factorization, and implicitly the two positivity constraints, actually it's huge sparsity constraint for our problem. And we started with L1 constraint. In the end, we didn't need it if we moved to sparse positivity constraint. And we're hoping that this, we call principal patterns, will play a role in a lot of bioinformatics tests as a basic decomposition like what PCA played in low dimensions for the decomposition. But of course, we have additivity so that we can look at things in the, based on like the heterogeneity and look at parts by parts. So that's the hope. As I said, when Ben approached me and kind of matchmaking me with Irvin Fries, and I was actually talking with my postdoc on computer vision, Julian Morel, about this problem. Ben approached me because he recognized, and more than I did actually, I like image data. I work on remote sensing images and computational neuroscience images, and that's why he approached me with this problem. So they said about no-code networks. And the first thing is, we've been working a lot, the first V1 for visual cortex is actually this Gabor-like wavelet decomposition. And this Gabor-like thing came out from data. It didn't come from mathematical formula. Sometimes we do use that. You have patches of images and do sparse coding. Those are the components that come up. These are like edge detectors. Okay, so that's why we borrowed the idea. Cross from computational neuroscience to statistics. I think that's the kind of the power of statistics is actually we borrow from and cross-fertilize. And we use that, actually worked really well because the different parts were recognized by Irvin and Sue like as meaningful biological parts. Later, we migrated to non-negative matrix factorization. And we get similar results, but this is just cleaner in practice. So our theory actually is still on the other version. So this is, remember I had 405 superpixels. You line them up as a big vector. Doesn't matter, you just have a fixed order. They have to match. But we didn't take into any spatial contiguity for this decomposition. Right, so you have, we have like 400 something vectors. And non-negative factorization says that we want to decompose it into columns of a dictionary matrix with a coefficient matrix. So you're basically postulating that each these gene images is an edited summation of these different parts and with different loading coefficients. And because I think the teams, the genes do work together respecting the physical boundary like pre-organ regions, you end up with spatially coherent subregions. And in higher dimensional, later state, we might have to put some spatial constraint, but this just fall out. We didn't put any constraint, spatial constraint here. I think it's because the way the genes work do respect these boundaries, because they're building things together to have different organs come out later. And this is earlier than morphology can help biologists to say, the different texture coming out. This is basically go beyond what the current human vision technology can see in terms of organs. This is what we call pre-organ regions. And we have positivity constraint on both the dictionary matrix and the coefficient matrix. This is all very high dimensions. So if you think about in dimension D, the positivity constraint give you 1/2 to the power of D, like fraction of the whole space. So it's pretty strong constraint. And therefore, that's why we didn't need L1 anymore on top of that. So positivity constraint, I worked when I was about labs, also on a network tomography problem. We started with EM and all kinds of things, and later we just use a positivity constraint and work really well. So it's a very strong constraint.
Can you explain why you said that this positivity constraint leads to sparse solution? What do you mean by sparse, and what is the connection between positivity and sparseness?
The positivity, to be exact, we have a lot of small coefficients for the A. So that's what I mean. And we later, I cheated a bit, we later did do another thresholding. Yeah. But you can directly do L1 on these two, right? You can add L1. And then, but just this very strong, give you a small coefficient. We did another round of lasso type of thing. Yeah. So the methodology contribution we made to this work is actually using stability but across different initial values. So for this problem, if you look at D and A together, it's non-convex. But if you condition A, D to optimize over D, that's convex, and for the other direction as well. So we randomly selected the original data as the starting value for the D. Right? So one big problem in a lot of this type of clustering, this is very clustering related, is how many dictionary you need or how many principal patterns you want to have. That's where we use stability. So you start with two random starting points, say 21 random images from your data. You start with them as D and just alternate and minimize. And then with some convergence criteria, you look at the final results and you allow permutation to match. So we use correlation. For each principal pattern you find for two runs, you find the best match, find the best correlated. And you do one minus that gives you the dissimilarity. If it's one, they are completely similar, you find your match. Okay, so this is you can call Amari type. Amari have worked on early things using this. And this is just make it symmetric. And then you look at that dissimilarity, you find 21 as the minimum. And you said 22 is pretty good, I agree. Actually, look at 22. One principal pattern got split into two. And we did a lot of stability analysis too. We followed everything through for 22 to make sure the conclusion don't change. So every step we made kind of arbitrary choice. We followed through a few steps to make sure that we're not just reading off that arbitrary choice. Yes?
So how is that different from doing the similarity on D8 and the whole product?
Yeah, we're looking at the D, not looking at the A.
And if you look at D8, would that be it's correlated, but is that...
Because we're not estimating DA, right? Here you go back to the data. Right? You try to decompose DA. Right? If you look at DA, that's fitted value. We need more than that. That will be a prediction. If you want to do prediction, that's a different game. But we want to interpret the Ds. So that's why I want the Ds to match.
Is the stability of DA and the stability of D? Yeah.
It's different. It's different. So you have to decide on the metric. For our interpretation, we want the individual Ds to be interpretable. That's why. So you could, if you want to do some prediction or smoothing, then you might want to look at that. Some other paper we did will actually look at that. So it's very problem specific how you devise your stability metric. Depends on the task. Yeah, good question. And later, actually, we learned that non-negative matrix factorization had been used in bioinformatics. You remember we came from computational neuroscience. And, but the way they used it was different. They usually do the dictionary and use it as features for clustering. And we're really interested in interpreting the dictionaries, but very much, And they didn't look at dictionary stability because their interest is for clustering. And they have something based on clustering for stability. Depends on the goal. And just to give you a sense, give you some confidence on what we do here is sensible, we did a simulation. It's a famous swimmer data simulation from one of the early papers to analyze non-negative matrix factorization mathematically, Donahoe and Studden. They have 16 generator patterns, like different limbs of a swimmer. And you put them together randomly, you get different swimmer postures. And of course, we nailed it down just like that, right? That's just to give you a sense that, so we did some theoretical work. Basically, there are two things important here. One thing is so-called the incoherence of the dictionary elements. Imagine that if D1, two columns on top of each other, there's no way you can separate them based on linear decomposition. But if there's only one of them, it's true, you have a chance to separate them, right? Because you don't need the component. So the sparsity and incoherence are both good. If orthogonal, because we're looking at the underdetermined case, you definitely have no problem separate them. A little bit correlation is okay as long as the sparsity is there to help you. So for us, the correlation, we're actually digging into it in simulations. And the theory is not exactly matching it with the practice, but we're going there. But basically, we're not too bad in early development stage because things are growing from different parts and not overlapping as hard as later. And later, there's a 3D problem because these little animals are 3D, and we're only looking at the flat view of projection. So that's what we got. With that stability-driven non-negative matrix factorization, we got 21 we call principal patterns. And I'm from mathematics as an undergraduate, so I loved it. It's like the vertical, the coordinate system, and the horizontal coordinate system. It's pretty amazing. But these ones, I just said was something else, right? But these two lines, I loved it. It's like X and Y, right? Telling where to go. And there's some odd bits, right? Not everything go that way, but the major part are pretty nicely lined up, vertical and horizontal. And then we did a very careful job. Really, this is a schematic map, but we did the best we can. We did them as much as an ellipsoid and put on top and map. That's why we have this half circle, because this one mapped kind of half into the gray. It's like a yolk and half into the mesoderm. Right, this is half foregut, and the purple is neurogenic region. Right, this is like stem cells, and this is posterior midgut. So you can see that all the early regions are food and shelter and some brain. Right, that's basically what's going on. You have a lot of skin development, you have the gut system developing, and beginning of neural systems. So then this was, well, we were here actually maybe 2 years ago, we already done that work. And the next question asked us, so first we wanted to do knockout experiments. And that just took a long time. So we had to publish this work before that completely finished. And we asked ourselves the question, how do we validate what we find? We like it, but is that enough? We look at the patterns, look meaningful. Can we push us harder to use it, give more evidence this is a useful decomposition? So the first thing, I don't remember exactly the order. One thing we end up doing is that we can characterize genes according to the different principal patterns. So you look at one region, and you can construct a correlation gene network. And 20 or 25% of the genes in Drosophila, even they know that genes are called the computational genes. So they don't know functionality. What? Oh, wow. So, and then we can do prediction, and then we can do local gene network construction. So let me go very fast. So this is an attempt to do that. We build different, see where they express themselves. So you can see this block, very much anterior, remember to the foregut, and this part, I think, related to the hindgut, if I remember correctly. So you can now give functionality, right? And 20% of the genes, this blue part, are computational genes. And you can also look at crosstalk, right? Whether they are close together, they share more genes for the different regions, and so forth. And this is go back to compare with pixels, and the thing Owen worked on sparse Bayesian factors, we get similar prediction result. But in terms of interpretation, the pixel-wise doesn't really interpret, just isolated bits. And the Bayes factors give negative kind of values. That's why, so altogether, we like what we have done because it's positive and also sparse and gives similar prediction. Okay, so let me just jump to the gene network. So we went back to the gap gene network. We have 40 pages of supplement material. So there's a lot of detailed work I'm not telling you because you have to map things together. We basically reproduce 11 out of the links for the gap gene network using our methodology. And for the, so the lab has been doing CRISPR-Cas9 experiments, and we end up with 40 experiments. And let me just show you some results. So we have to also do which is upstream, which is downstream. And this is knocking out a particular computational gene. The number is pretty big. And this is a wild type for this PERU gene called FTZ. And this is the wild type. This is mutant. And you can see that the head is elongated. And we have done a lot of work trying to put it together. So the wild type is the purple, and you have a little darker green is the overlap, and you have the mutant. Seem to suggest that on the population level, there is bigger, longer head span for the mutant. And there's also some evidence we know this gene is definitely important because before hatching, about 16 hours, all the embryos die. But we're told you cannot publish paper said because they died. You have to say why it died. And it looks like, it seems the neural system, the nervous cord is messed up. And we're in the middle of doing more experiments with Sue's lab and try to really prove the case that we found the gene is important. And we tried on RNA-seq. So you can see the principal patterns. It seems to be meaningful too, but we haven't really gone further than that. And we like to be able to try, I think Church Group about 2 years ago now, supposed to have a technology which can simultaneously measure gene expressions for thousands of genes. And that will be great because we won't have matching problem. And we like to be able to contribute to human genetics, if anything, funding agency wants you to do that. And it's supported by different organizations. And this is the paper just appeared online at PNS. Thank you.
Sure, I can start.
Yes.
So as you said, the work is pretty similar to what we did a few years ago in terms of the idea of decomposing these spatial patterns in unsupervised fashion. So what we did at the time was to put the sparsity on the factor loadings with the idea that each gene will only be controlled by a small number of factors because hopefully these factors represent positive and negative transcription factors.
Yeah.
So you don't have a sparsity constraint like that, right?
The early version we did sparse coding is very similar. And we find that with just the two positivity constraints give very similar result. Well, first version from computational neuroscience was very similar. You kind of took the Bayesian version.
Right. Exactly.
We didn't quite know when we started. Actually, a word later we discovered, at least the people, maybe Irvin knew, but maybe mathematically he didn't realize.
I mean,
But later we definitely recognized that, and that's why we compared. Yeah.
It's slightly different versions of the decomposition, obviously. The other thing that we noticed.
But I think it's the interpretation part. I think that's where we differ. We part our way a bit. Yeah.
The other thing that we found helpful, and that's what people also seen when they used, we already stole this idea, of course, from other people before us. And so from expression analysis, for instance, when you do these decompositions, often you end up with factors that are really useful because they represent your experimental artifacts and biases. Like, for instance, we had some that represented just, blurry outlines of the embryo because some of the pictures were out of focus, or you would have lighting differences where the top was always a bit lighter than the bottom. And so we would have these factors that soaked up the artifacts.
Yeah.
So did you also see that? Because we found that pretty helpful.
I think we did a lot of screening probably for the image before we went in, right? Yeah, so we probably avoided that because we really want the interpretation and for the local gene network. I agree the beginning part very similar, but then our goal was more not prediction. We did a prediction to compare, but it's more like to build local gene network, yeah. But I think we did a lot of manual human screening for those bad images.
Yeah, because we threw everything at it and the kitchen sink, so we didn't even filter out whether the embryos were in the pre-processing and the correct orientation and everything. It was
We did pretty careful pre-screening. I think if we didn't, we'll see similar thing. We're probably going to have very weird parts. I'll believe that. Yeah, and there's also like de-blurring, all that stuff. Yeah.
So did you the input of your algorithm are just 6 gapped genes, right?
No. We started with 405 genes. I just concentrated to compare with the gap gene network. Yeah, we didn't just use that. We have lots of computational genes, like 20% was the computational genes. Yeah.
Thanks. Good, thank you.